Hello and welcome back to episode 5 of Callum the Biologist with me, Callum, a National 5 biologist in the Highlands of Scotland. In today's video we're going to speak all about proteins, and this is from the National 5 Biology Unit 1 course, which has got cell structure, the transport across cell membranes, DNA and the production of proteins, and then today's episode is all about proteins. We've got 2 more episodes to complete this unit. So keep your eyes out for episodes 7 and 8. Before we start today's video, I just want to make you all aware that I have got myself an Instagram account for biology. So it's Callum the Biologist on Instagram. So please head over there and give it a little follow. So first of all, like every single episode that we've spoken about in the National 5 unit, we have got a keywords list. So we have got an enzyme, which is the biological catalyst made by all living cells. A catalyst is something that speeds up a chemical reaction without using up itself. The active site is the part of the enzyme that binds to the chemical substrate. The word complementary is a key word for this unit, and it'll be used quite a lot when you're speaking about complementary base layers, etc. And this just means it fits together. So the active site is complementary to the substrate, and the substrate is a molecule at the start of the reaction. The product is the molecule produced at the end of the reaction. Degradation. How do I say that? Degradation. Let's just go with that. And synthesis. Synthesis is the build up, and degradation is the breakdown. Enzyme substrate complex is the term that is used when the enzyme has binded to the substrate. Optimum conditions is the conditions that allow the enzyme to be at its most active stage. And denatured is another word that we're going to be using in this unit, and mainly if you're a biology student, this is where you can use it. I don't think we're going to speak about it in today's video, but you will if you're undergoing a National 5 course. So denatured is just when the enzyme changes its shape and the rate of reaction decreases. So there are millions of different types of proteins in living things on planet Earth. Different sequences of amino acids allow different proteins to be made. But what are proteins needed for? Some functions in the human body can be remembered by the acronym SHARE. So this 5-letter word can be used just to remember the functions of proteins. So S is for structural, H is for hormones, A is for antibodies, and that fits in very well with the coronavirus unit. R is for receptors, and E is for enzymes. So enzymes are biological catalysts, and they're used to help speed up the rate of a chemical reaction in cells without using up themselves. Enzymes are made from proteins, and their shape is very important to how this affects their role. The shape of the active site of an enzyme is complementary to the substrate, and this means that each enzyme will only work with one type of reaction. So step 1 of this process, the enzyme binds to the substrate at the active site. Step 2 is an enzyme substrate complex is formed. Stage 3, products are formed, and these are released into the enzymes and can be used over and over again, and this does not use up itself. So there are 2 types of enzyme reactions. We've got degradation reaction, which is the breakdown reaction, and the substrate is one large molecule and the product is 2 or more smaller molecules. So hydrogen peroxide is the substrate in this example, and the catalase is the enzyme. Oxygen and hydrogen are the products that are made. In a synthesis reaction, this is the build-up, and the substrate is 2 or more smaller molecules, and the product is the larger one. So amino acids are the substrate, the enzyme is in the middle bit, and the protein is the product at the end. So what happens when you change the temperature or the pH? Enzymes are most active at their optimum conditions, so this is an increase in temperature or a high or low pH that causes this protein to lose their shape. The protein is said to be denatured. So this is the loss or change of shape, and the rate of reaction is decreased. If this happens in enzyme, then the rate of reaction will be lower because the active site will no longer fit the substrate. By testing enzymes at different temperatures and pHs, a graph can be drawn to work out these optimum conditions. So, that is all we're going to speak about in today's video, but we are going to briefly just speak over some exam questions. I don't have them up on the screen, we're just going to talk through them. If you'd like to see some past paper questions, you can go to sqa.org.uk/pastpapers/findpastpaper.htm !subject equals biology and level equals national 5. So, questions that you might be asking is describe an enzyme action, and this could be worth 3 marks, and the answer you'd be looking for would need to have 3 key sentences, and it need to have a lot of scientific information for this. So your answer could be: an enzyme is a biological catalyst. Enzymes bind with their substrates at their active site. The active site has complementary shape to the substrate. This allows them to fit together. An enzyme substrate complex forms and the reaction takes place. Products are formed and released by the enzyme, and the enzyme can now be used again.